Okay, we now come to the final talk of the whole conference, and by no means least, however, is my pleasure to invite Dr. Isioma Egbuniwe from University of Nottingham, whose talk is entitled Ultra-High Resolution Mapping of Spatial Patterns of Gene and Protein Expression in Cholangiocarcinoma and the Cholangiocarcinoma Tumour Environment. And this is, like the last talk, has been funded by AMMF.
Thank you, Shahid. Hello, everyone. First of all, I'd like to thank the AMMF for funding this work and for inviting me to speak. And thank you to all of you who are still here and are still awake. I promise I'm not going to keep you for long. This work is a collaboration between myself and Professor Kevin Gaston and Professor Sheila Jaya-Raymond at the University of Nottingham. I think we started this maybe last year, so it's still in its very early phases, and I'm just going to give an update of where we are and how we've sort of overcome problems, quote unquote, along the way. So I'm an academic histopathology trainee or registrar in the last year of my training, and I've just come off an NIHR-funded clinical lectureship. So in addition to being interested in HPB pathology, in my former life, I was also a researcher in cancer immunology. So looking at the tumor microenvironments marries those two great interests for me. So just before I start, I just want to highlight other AMMF-funded work that's going on at the University of Nottingham. So there is a project that's being led by Miss Grace Martin, who's a PhD student supervised by Kevin and Sheila. And this is looking at identifying off-patent repurposed drugs and novel drug combinations that have the potential for treatment in cholangiocarcinoma. And the MMF has also kindly funded another project that I'm leading on, which is investigating the role of the cytokine interleukin-17 in tumor lymphangiogenesis in cholangiocarcinoma. So this is our lovely campus, which I unfortunately don't get a lot of time to visit because I'm stuck in the basement like Tim, looking at slides. So the background to this project really started off with work that Kevin and Sheila had previously done, looking at this, looking at proline-rich homeodomain, which I'll refer to as PRH going forward. And this is a transcription factor that's really important has been shown to have a role in different types of cancers, but is also important in proper development of the liver. And they had shown from some work that was published, I think in 2020, that PRH was overexpressed and drives tumor growth in cholangiocarcinoma. So on the left panel here, using immunohistochemistry with a commercially available patient tissue, they stained up cholangiocarcinoma tumors on the right and compared that to staining of non-tumor bile ducts on the left. And when they quantified that, they could see that the average intensity of staining for the PRH transcription factor was a lot higher in the cholangiocarcinomas than in the non-tumor bile ducts. And they went on and further looked at this in different cell lines by Western blotting, I believe. And that the data from the patient tissue was borne out in the cell lines. And when they took this into a mouse model where they looked at tumor growth in control animals, so these control animals had, let's call it normal PRH in the blue line, compared to knockdown PRH in the red line, they showed that in the mice that had little to no PRH, you had the tumors were much smaller and did not grow as fast. So going on from that work, or rather parallel to that work, in my work looking at cytokine secretion in cholangiocarcinoma tumors, I've been interested in using a type of analytical tool called the Vector Imaging Analysis System. And what this allows you to do is to stain up to 7 colors, multiplex immunohistochemistry or immunofluorescence imaging. And this image I'm showing is a whole slide scan of a cholangiocarcinoma tumor, and it's lit up quite prettily like a Christmas tree. But the different colors represent different compartments within the tumor. So in this scan, you can look at T cells, macrophages, which are parts of the immune system. You can look at tumor cells. You can look at blood vessels and lymphatic vessels. So having used this technology in my own project, looking at cytokine secreting T cells, I had I don't want to call it a level of, I should call it a level of expertise in analyzing this kind of data. So quite fortunately, we had, I think about 4 years ago now, started off a preliminary project using this newish technology, which is nanostring digital spatial profiling. And in the overall idea is similar to the vector imaging, but what nanostring has in addition is it allows you to look at, while vector imaging allows you to look at 7 proteins, nanostring allows you to look at upwards of 100 proteins and even genes. And it's described as spatially resolved technology, which means that you have the slide, basically, as we would look at histologically. You can look at different proteins and genes in situ, so basically without breaking up the tissue. And it's quite nice because you can look at the way genes and proteins are expressed in native parts of the tissue. So you can look at gene expressions in glands, non-tumorous glands versus tumorous glands. You can look at gene expression in the blood vessels, in the lymphatics. So this is just an example of the type of staining we did, looking at 2 cholangiocarcinoma tumor samples. And in green, we have the pancytokeratin stain, which identifies the tumor cells. Yellow is smooth muscle actin, which is the stromal cells, sometimes referred to as CAFs, as we heard about earlier. In the red are the CD3 positive T cells, which are immune cells. And blue is a nuclear stain, which essentially just helps you to be sure that what you're looking at is a true cell and not just background. And after the staining, what we can then do is select what we term ROIs, and these ROIs stand for regions of interest. So based on what you've stained for, you can choose to interrogate different parts of the tumor. We heard about immune cold and immune hot areas of tumors, and immune cold basically means for whatever reason, immune cells are not able to infiltrate into that part of the tumor. You can select, for example, an immune cold tumor, an immune cold region, I beg your pardon, where immune cells are excluded from, and probe that area further to look for proteins and genes within that area, and you can compare that to a next door immune-rich area where you have lots of immune cells in there, and that will give you a spatially resolved picture of what's going on within the tissue. So this is just some further analysis, some further downstream analysis from what we'd done in this preliminary work 4 years ago. By looking at different regions of interest, we were able to corroborate that there was a differential presence of CD68 macrophages, which are thought to be they're part of the immune system, but sometimes they can also act like tumor stromal cells. So we showed that within different regions of the tumor, you had differential accumulation of macrophages, and we could generate different gene signatures from those different areas based around the macrophages and look at whether different immune pathways were upregulated or downregulated within those areas of the tissue. So that was why Sheila, Kevin, and I came together to say, and we put in an application to the AMMF saying we have this technology, we have this expertise. we would like to look at cholangiocarcinoma tumors to sort of interrogate what's going on within the tumor microenvironment. So the next step was creating tissue microarrays with intrahepatic and extrahepatic cholangiocarcinoma tissue. One of the reasons I love this meeting is you get to hear about not just what work is going on elsewhere, but what resources are available. Because a lot of the time, as researchers, we're sort of siloed. And you think we talk to each other a lot, but sometimes we really don't. And this was where we sort of hit our first roadblock, because getting access to tissue was quite difficult within our local center. And what I then decided to do was sort of piggyback off of, after asking lots of questions, I started a collaboration with another group who had ethical approval to do similar work, but in pancreatic cancer. But they had ethics to look at tumor tissue from the hepatobiliary tract, including the pancreas. So they had bile duct and liver tissue. So after discussing with the investigators on that project, I was allowed to put in an amendment to their ethics, which took quite a while to go through. But thankfully, we eventually got the green light. And what that meant was that I was able to retrieve cholangiocarcinoma patient samples. These are the paraffin-embedded blocks and the H&E slides that were resected at Nottingham University hospitals between 2003 and 2023. Tim earlier talked about the storage in Wales. So I had the wonderful job of reviewing every block and slide from over the past 20 years, including those retrieved from Wales. Took a lot of time. So all slides and blocks were reviewed for quality assurance by myself and a second independent consultant pathologist. We identified 23 intrahepatic cholangiocarcinoma samples, so both matched tumor and non-tumor tissue from the same patient, and 42 distal bile duct samples. And the reason this number might seem so low is in selecting tissue, we were very careful. So first of all, we didn't select biopsies, because biopsies contain really small amounts of tissue. And as Tim said earlier, we have to have some of that material in the block in case of future molecular testing. So we only selected cases where we had enough tissue to use for research material without compromising the rest of the diagnostic material that was still left behind. And we only selected cases where we felt we could get good quality tissue from the block. So after all that selection, then we went through all the clinical data and updated the clinical database. Now this included updating the tumor stage. So in pathology, we use something called the TNM staging system, which stands for tumor nodes and metastasis. And some of these some of the patients were seen when the staging system was at stage 5. And things changed, the iterations are updated, so we're currently at TNM 8. So part of what I did was to upgrade our database for every single tumor to TNM 8 and update the clinical data as well. So this is a hugely useful resource that we now have. So just some of the cohort data from the intrahepatic cholangios. The median age was 64 and a half years. The most frequent age we saw in our cohort was 64 years. Quite surprised, there was one outlier who was 27 years old. And in terms of the gender split, we had slightly more females in our cohort. The majority of tumors were diagnosed, were of an intermediate grade. Very few were low-grade tumors. LVI stands for lymphovascular invasion, PNI is perineural invasion, and RM is resection margin involvement. Now, these are things we look for when we're reporting tumor cases histologically, because they correlate with unfortunately poor prognosis. And what we saw was the majority, over 70%, at the time of surgery, had one or more features that meant they were likely to do worse. And also we looked at background, we also collected data on what the background of these patients were. From a small intrahepatic cholangiocarcinoma cohort, just over 10% of them had background PSC, which is quite high. But this ties into what we've heard over the past few days in this conference. So the next step, what we wanted to do next was to optimize our antibodies using patient tissue. Now, PRH that we're interested in is a commercially available antibody, but Kevin and Sheila are also interested in phosphorylated PRH, which is the active form, and this was formulated in-house by their groups. So we wanted to be sure before going ahead to do the nanostring analysis that these antibodies would work, not in cell lines, but in patient tissue, because that's what we were going to be doing our final downstream analysis in. And this paper was published, which showed, published in 2022, which showed a potential role for, so PRH is also known as HX, a potential role for PRH or HX in colorectal tumors. And we had access to colorectal tumors through some of our collaborators at Nottingham. So working with our collaborators at Nottingham and with an expert in nanostring analysis, Professor Sergio Rutella and Dr. Jay at Nottingham Trent University, we started working to optimize these antibodies. So I don't know if you can see this, if this projects clearly, but what you've got, if you look at these little, they sort of look like ovals, but they're not all, they don't all have nice rounded edges. So these represent malignant glands or colorectal cancer tumors within the microenvironment. And what Jay has very nicely done is to optimize the staining for PRH, which is in red, and the phosphorylated version, which is in green. Sorry, the phosphorylated version, which is in yellow. And there's some areas that look sort of orangey, and those represent where you have both PRH and phosphorylated PRH within the tumor, which is the tumor glands are highlighted by the green. So any orange color we see must be within the green to show that it's real staining. So we were happy that this worked in colorectal tumors. And then Jay went on to try and optimize this within our CCA tumor tissue. I apologize, this doesn't project quite well. So what I've done is I've split what the channels we're interested in into single color channels. So in blue on the left, on the far left, we have the nuclear stain. In green, we have cholangiocarcinoma tumors. In red, we have our PRH transcription factor that we're interested in. And in yellow, we have the phosphorylated PRH. And when we put that together, we can see that we have true staining. Again, like the colorectal tumors, if I was going to try and draw around this outline, this irregular ovoid or oval outline we kind of see is a cholangiocarcinoma tumor gland. And we can see that we have some areas within there which are orange, some not so much. But this is what we expect, because we don't expect, we expect differential staining in different parts of the gland for our phosphorylated PRH and the primary PRH transcription factor. So thankfully this works. We've done different iterations of this over and over again, and we're happy that the staining is doing what it says on the tin. So what we're doing next is, actually just after we got the staining working, the machine broke down. It's such a highly specialized machine that it's taken about a month to get fixed, but we've hoped we would have started the first intrahepatic CCA array of 23 patients and matched non-patient tissue should hopefully have been stained this week. So fingers crossed we can move ahead. So just to talk about what we're doing. So like I say, Jay has stained our intrahepatic tumors, and we're all going to sit down and figure out what regions of interest we want to look at. So we are going to be looking at PRH protein and phosphorylated PRH, and quite nicely we can look at immune cell infiltration within areas of PRH expression. I didn't talk about another project we have running that's kindly funded by the BMA Research Foundation, because we have some preliminary data that shows that PRH potentially acts synergistically with T cells within cholangiocarcinoma. And in that project, we're looking at the cytokine signals that the T cells are releasing. And the questions we're asking are whether PRH and T cells are reciprocally regulating each other. So quite nicely, we can test some of our ideas in this model, because we can then look at immune cell-rich areas in the context of phosphorylated PRH expression and non-phosphorylated PRH expression. So we're going to do that, selecting regions of interest. And then with the downstream analysis, we're going to be using the nanostring human immuno-oncology atlas. And that allows us to look at 570 proteins within this patient tissue. And we can select up to 10 regions of interest. So that's 570 proteins times 10. And that's one of the reasons we're collaborating with Professor Rotella, because he's one of the UK experts in this, the analysis of this type of data. Because the potential problem you have is you have so much data, and if your analysis pipeline is faulty, unfortunately the data ends up not being useful. Yeah, so that's me. So in conclusion, I've sort of showed you, I've talked about PRH and the fact that Professors Gaston and Jaya Raymond have shown previously that it's overexpressed in cholangiocarcinoma tumors and drives tumor growth. We've shown that we can apply digital spatial profiling based on immune cell distribution within cholangiocarcinoma tumors. We've created this great resource of intrahepatic and distal cholangiocarcinoma patient tumor microarrays with matched pathological and clinical follow-up data going as far back as 2003. And we've now optimized both of our antibodies in 2 human systems, so both the colorectal cancer and the cholangiocarcinoma patient tissue. And now we're going to be working on selecting the regions of interest in further downstream analysis. So watch this space. I'd just like to thank all our collaborators from the University of Nottingham and Nottingham University Hospitals, Professor Dilip Lobo and Dr. Abed Zaitoon. Dilip is a HBB surgeon and Abed is a consultant HBB pathologist. They've been really helpful in letting me use their ethics, and Abed has been really great in looking at the pathology slides with me as well. Our collaborators from Nottingham Trent University, and our collaborators from NanoString US and Kelly, who was initially at the University of Birmingham but is now at ProPath. They helped us with the initial cholangiocarcinoma tumor analysis in 2024. And thank you to the AMMF and the BMA Foundation for funding. Thank you for listening.